So today we begin to discuss the prediction of subcellular localization, and this begins to somehow bring the central part of this lecture, which essentially is about throwing everything you can at the problem of prediction at hand, and in this particular case, subcellular localization. So here are three different words, or three different terms. What do you associate with them? Let's begin with the first one. A prediction by homology. What is that?
When you search for similar sequences, for example, or similar structures, and then you derive information based on the most similar ones.
What do you know what homology means?
From the same ancestor, or?
Typically that's what it means. So why is a similar sequence from the same ancestor, or why do you infer that, or why is that used? Because somehow the idea is that the reason why you can infer from sequence similarity in function is because they're evolutionarily related. And for every single time you apply homology, you in fact have to have another protein for which you have an experimental annotation. And then you say, okay, this one is similar in sequence to that one, to my left hand. And because of that similarity, I say it is an ADH. All right? So whenever you do the inference, at the moment of the inference, you need the ADH. You need to say this one. That is the meta-student idea. You find the one in your data set that is most similar to it, or the set, and then you say, because there I have an experiment, because of that sequence similarity, I make that deduction. Now, let's look at the other, map it out, and call this the other extreme end of it, ab initio. Do you have any association with that term? So the idea really is, one way of translating that in physics is from first principle. Okay, what could that be then?
You have no reference sequences or something, and you predict without this information. What
would you use for the prediction then? So you cannot not have nothing. So you need something. So what do you have? That's a good start. Let's do the next step.
Yeah, maybe you know one amino acid is responsible for anything, and then you predict with this. So
you say that we know XE forms a beta strand, to get back to the discussion you had before on secondary structure, or something like that. And then you string it together and you say, I have some sort of ideas or some biophysical concepts, and according to these biophysical concepts, I predict secondary structure. Okay, but now we are looking up a feature. Would that be ab initio? Or would it be de novo? Would that be sort of from first principles? So this is a borderline case. In some sense, first principles for a physicist imply that you write down an equation. You can sort of write down an equation what you have an amino acid that gives a certain hydrophobicity. You can possibly even measure that from the chemical structure. And you somehow can write an equation that would then essentially predict secondary structure. So in some sense, this is almost like ab initio, not quite. There's something here really important. De novo has always this machine. One way of explaining it quickly is to say machine learning devices operate de novo. They don't base on first principle. It's not writing an equation. You train machine learning device from knowledge you have. Why is that not homology? Because at the moment of the inference, you predict that this particular protein is an ADH without having one particular one that is the ADH that you relate to it. Yes, you trained on many other proteins that were all enzymes. And from these enzymes you extracted some knowledge in a machine learning device. But it does not allow you the one-on-one. The moment you predict, you may not be able to say this is because that protein is annotated. Homology does exactly that. You do the inference by homology because of one particular protein that you can name and show, and you can get the literature reference why that is called an ADH. For the machine learning device, that's this statement about black boxes. The black box extracted some rules. The moment you make a prediction, it may not be quite clear where that rule came from. The rule came from the training over a bunch of proteins, right? And that typically is sort of the distinction between these. Now, let's get into the problem again of subcellular localization. So, on the one end of the problem, you have essentially the bag in a prokaryotic cell where you have no subcellular compartment. You essentially have an outside space, an inside space, you have a membrane, and everything is sort of distributed in there. The DNA is distributed all over the place, and every intruder is somehow in there. It's just one big bag that does it all. Now, since some operations are, for instance, proteins that interact with DNA, in the situation of the prokaryote, when you want to interact with DNA, you have to make sure that the protein that is close to the DNA, in fact, is A, interacting with it, B, doing the right thing, and C, that there's no other protein that interacts with DNA that comes to the same location. Since DNA is all over the cell, it's not that easy to regulate, and it poses a problem. Now imagine you could put all the DNA into the core, into the nucleus, and then all you need to do is put everything that interacts with DNA in there. Now you still have to sort of coordinate the action, but you already simplified some story. So you could, for instance, by just leaving some DNA interacting enzymes in the mitochondria, you could already separate the way you do it. So the ones that operate on mitochondrial DNA are not in the nucleus, so you already, by separating them into two different compartments, completely simplified the task. Now, in this context, the task becomes, how do you get the proteins move around? How do you make sure that the ones that go here are really the ones that interact with the DNA? And that is essentially the sorting problem. Another way of looking at this, why do we look at subcellular localization at all? Well, subcellular localization is one aspect of function. Because if you know it's nuclear, you know something has, almost all the proteins in the nucleus have something to do with DNA processing. Nailing it, you don't know how that enzyme works. You don't know which pathway it works on. You don't know what the disease has to do, but you do know it has to do something with processing of DNA. Okay? It's not a very high resolution prediction of function, but if you know the protein is nuclear, you know something about it. Okay? That's the first reason why subcellular localization is interesting. The second reason is, when you look at EC numbers, there's a hierarchy of numbers. When you could look at GO numbers, there's a hierarchy of numbers. And all of those are assignments that essentially are done by people, and they make sense. Localization is something that you can measure. It may not be the most informative thing, but it's very objective. Okay? You have compartments, and you can either say it's in there or not, or it travels. But it's something that's very, not easy to measure maybe, but it's very objective to measure. Whether something belongs to that enzymatic class or to that one, there are different ways of looking at it. So in some sense, it's a very objective function. Now, this is one way of looking at subcellular localization, so high resolution. You have the different types of compartments. There's another way, a more detailed way, where you have sub-compartments, where inside of the nucleus you have the nucleolus, you have a part that is close to the membrane, the matrix, there's an extracellular matrix. There are all kinds of sub-compartments. And then there are compartments that are temporal, in the sense that for some time you have a bunch of proteins, sometimes hundreds, that come together and form some structure that is important for some role, okay? And you may call them compartments because they exist over a certain period of time, and they clearly have a functional assignment, but that gets sort of blurry. On this level, it's very clear because you have essentially whatever I show here is surrounded by membranes, by lipids. And on this level it's even simpler, where we essentially have cytoplasmic, nuclear, and extracellular space. Now, again, this is the same thing. There is a hierarchy of sorting mechanisms. The blue ones is essentially vesicular transport, so everything that is going toward, most of the proteins that go toward secretions leaving the cell, they are transported from the ribosomes to the outside of the cell by budding to different sort of vesicle systems. That could be the most often taken path for proteins that are secreted, so that get out, is you go through the endoplasmic reticulum, through the Golgi, typically some other vesicles, and then you're secreted. Means put out. Some proteins end up in the membrane, they sit in the membrane, and some get out completely. Now, the types of methods we can use to predict subcellular localization: sequence similarity, text similarity, motifs, and today we are going to go to some of this level. Let's see how far we go. The first one we already touched, homology-based inference. Again, homology means that I have another protein for which I know the subcellular localization. I look up sequence similarity and I can infer it. When they are very similar, I can do it. That's the story from Rajesh, where we take all the proteins of known subcellular localization, take a unique subset, then essentially work on a function that infers from sequence. One function is percentage sequence identity, shown here. No, this is the alignment length, the percentage sequence identity, and then you have a curve like this one here that somehow fits the red curves of similar red points of similar subcellular localization and the green of non-similar subcellular localization. Again, this line somehow fits it. Some line above this somehow fits it too. That is called the h-val, where the h-val is simply the distance from here. As all repetition. Again, here is just visually the same thing. So that's the curve that I showed you before, and this is a distance plus 5 plus 10, blah, blah, blah. And down there is minus. Okay, the curve that you need in order to apply different, for different subcellular localization classes, the curves are slightly different. So this is accuracy versus this h-val, the HSP distance here. Is there anything else we can use to measure sequence similarity? Again, percentage sequence identity, this h-val. Anything else we could use to measure sequence identity? Oh, sorry, sequence similarity. What's the most common way in which you measure the similarity between two sequences that you all have encountered most in your studies? I would challenge.
To blast.
Yeah, so how does a blast do it? How does a blast actually sort its hits?
With an e-value.
What's the idea behind the e-value?
Sorry?
So somebody else, so far so good. Anybody can explain me briefly what the idea behind an e-value is?
The expected number of sequences we should find due to the size of the database. So
essentially the idea behind that is that you look at the similarity between two sequences, and you know that for a certain sequence there's a certain random probability of matching it, and what you want to see is how the score that you observe differs from the score you would observe at random. So essentially it is this model here where you have a random background, you have a finding, you look up, and that again, the value you look up here could be, in principle, sequence identity. This could be anything. In this particular case, it's just the way the algorithm operates. It's a different score, but in principle it could be that. And for any kind of score, again, PIDE could be the one, you now want to have an e-value that essentially relates two aspects. It's written, two aspects are important for the e-value. One is the distance between the value that you observe and the average here. And what's the other one that is important for the e-value? So the further it's away, the more significant the hit obviously is. But there's another aspect of that distribution that is relevant here. One, the distribution, let's assume a Gaussian. What are the two values that characterize a Gaussian distribution? Average is 1.
Standard deviation.
Yeah, exactly. So the width. So just imagine a distribution that were much wider. If the standard deviation would completely be such that, you know, it would essentially go like this. So this would be entirely in the Gaussian background. then the average may still be here. Okay, okay. Are you arguing against me because we hit 0 and then this can no longer be a Gaussian? It would be, I misread your... So one point of criticism, of course, to my argument is it's a Gaussian, it's limited at 0, it cannot be that big. Okay, okay, okay. So my argument is a little bit pushed here. But you see my point, right? So you just have to move the point here and then I can still. So imagine the red one would be here and then all my argument does absolutely work, even with critical. It's not what you meant. Okay, so here, this point stays here. Anyway, those are the 2 points. That is the e-value, and it's another way of measuring it, and essentially those 3 ways are what you have to assess. The way we assess them is we essentially ask, given a certain threshold, what is accuracy, what is coverage, accuracy and coverage. So positive predicted here, positive predicted, negative predicted. So positive predicted would be, for instance, nuclear. So I look at proteins being nuclear and not nuclear. Positive predicted would be the ones that are predicted to be nuclear. Negative predicted means the ones that are predicted to be non-nuclear. Positive observed means the ones I observed to be nuclear. Negative observed, the ones I observed not to be nuclear. TP, true positives, and so forth and so on. You know all of this. This is essentially a 2-state accuracy. We simply ask, of all the nuclear or non-nuclear, how many are correctly predicted? It's 2-state because nuclear, not nuclear is 2-state. Then there's accuracy and specificity is essentially of all the ones that you predict, how many are right. Coverage of all the ones that you observe, how many did you get at that threshold. And again, there are many, many different words out there. Coverage, sensitivity, recall is typically used for that. Then there is an asymmetry here. There's positives. So in this particular case, for the example that I gave, it would be nuclear. There's negatives for all the proteins that are not nuclear. And you unfortunately always have to look at all of them. This original, so one way of compiling all these numbers here is if you just started with the numbers. Entering the number here would allow you to compile all these numbers here. But nobody understands the matrix with numbers. So you can, our intuition can handle 90% accuracy versus 50. But looking at these numbers, at least the way I look at tables like that, I translate it exactly to those maps. So unfortunately we need those, and unfortunately when we need those, we typically blow up into again another set of 4 numbers, or even 5. And unfortunately, very often we need them all. The term of the wording here, so some parts of Wikipedia call only this one specificity. Some call this a specificity. Again, so the literature is very confusing. It's very clear what it is. Don't let yourself be confused. Now again, for this particular problem, we have to, in this particular case, look for instance H-val, percentage sequence identity, E-value. Here I look at BLAST, here pairwise alignments, here look at Psi-BLAST, profile-based alignments. And suddenly you have a very simple question, how well is sub-cellular localization conserved? And you get a bunch of answers and it's very confusing. Now, in this one you clearly see that Psi-BLAST performs better. The profile-based alignments performs better than the pairwise alignments. Did you? Did you see that? So why can you not see that? Because, in fact, the point is that I have 2 bunches of curves, and I did not introduce them today. I presented these curves before, so I'm rushing through it. There's accuracy versus coverage. And the points at which they intersect are different. What I show here is the threshold. Here it's the H-val, here's the percentage sequence identity, it's the E-value, the Psi-BLAST E-value, or BLAST E-value in this particular case. So we cannot put one threshold and decide whether one is better or the other, because we have actually 2 values that we have to compare, coverage and accuracy. What is better, 50% accuracy and 60% coverage, or 60% accuracy and 40% coverage? And there's no direct answer, unless you introduce an F-score or something like that. How can we compare the accuracy versus coverage curves? Well, one is we simply look at one particular point. And that is shown here in some sense, where I look only at accuracy and I pick out points. Another way of comparing them is by putting them onto the accuracy coverage curve. So on this curve, and then on this curve I can compare different solutions. On this curve I can see that the dark lines, again H-val, PID, E-val, gray is the same for pairwise, the gray are always lower. So here you immediately see it. But it's a long way, it's a lot of numbers. And unfortunately, very, very often you have in your thesis to look at all of these numbers. But you have to find a way to present as few as possible to yourself when you summarize the abstract, and to your supervisor and when you talk about the message. And see whether there is a way to use one measure, one number that explains it, and see whether that explains it or whether you're wrong and maybe there's something else to be said. Okay? In this particular case, I just gloss over a long story, you cannot follow me on that, but the black essentially is always higher than the gray. And then there's a point to be made that the H-val is above the others. But this is not important. Now, assume this gives us a threshold at which we say, okay, we can infer sub-cellular localization at 90%. That's good enough for me. I'm going to use the threshold. In this particular case, it's 40, H-val of 40. So now I publish a paper that says, by homology inference gives me 40%. At this threshold, it gives me an accuracy of 90%. I've assessed it. I've analyzed it by pairwise blast, by all kinds of measures, so I've collected a lot of data. Now, is that enough? Is there another thing you would put into your manuscript? Another thing you would look at this point? So again, let me back up and explain again what I'm trying to go after. We want to predict sub-cellular localization. One way of doing that is by homology. First on our assignment task, right? And here I now assessed by threshold. In fact, assessed 3 different thresholds, E-val, PIDE, H-val. I assessed 2 different alignment strategies, pairwise blast, psi-blast. I come up with an assessment of my threshold. Or is there anything else you would do? That's, in some sense, if you want, is the moment where the biology starts.